Okay, so welcome to the Click Seminar series. This is the third out of the 6 talks that we are going to give over the next weeks or month. Today, Akul Mehta will be talking about his GLAD software and how you can use it to analyze and visualize glycan array data. Akul is a member of Rick Cummings' group at the Harvard Medical School in Boston. And you actually heard him already speak on the first seminar where he gave the introduction into glycan arrays and also the experimental part of glycan arrays. And I said it before, he is really a person that does everything related to glycan arrays, starting from printing his own arrays, running own array experiments, and then analyzing the experiments and publishing them. Plus, in addition, he actually writes his own software. And that's what he is gonna show us today. Okay, Akul, please take it away.
Thanks, Renee, for the nice introduction. So, hello, everyone, and thanks for coming to my talk. So there will be a few slides where I'll just give you an introduction for what is this software which I've built, which is called GLAD, and how we can use it for exploratory glycan microarray data analysis. Here's my email address, aymehta@bidmc.harvard.edu. In case anybody has any questions, I will leave it in the chat at the end of the talk for anybody who wants to get in touch. And then there are just a few slides and then we'll move to a live demo, because I think seeing the software work in real life will probably help more people understand how they can use GLAD for their own applications. So just to give you a background, this workflow of the array experiment we kind of covered in the first seminar of the seminar series. And at the end of the array experiment, we usually get an Excel file which has like bar charts. So glycan array data is usually processed using Excel or other statistical software, and you get a list of glycans with their sequences along with the signal intensity for these glycans. And then this usually in Excel would be plotted in such a manner where you would have 600 glycans with their intensity values. Excel is great, but it does have problems with respect to when you're trying to visualize the structures of the glycans alongside such data sets, it becomes really challenging. It also becomes difficult to compare, multiple of these array experiments within Excel, because the plotting ability of Excel is kind of limited, I would say. And also the filtering and sorting functionality is also limited in Excel, because we use very unique nomenclatures in glycans, and that becomes challenging for Excel to kind of identify and filter and sort. So to overcome all these hurdles, I started developing this software called Glycan Array Dashboard. It is the main aim of GLAD was to make visualization and comparison of this microarray data simpler. So the way it works is, once you have a number of experiments, you could have multiple files, and you can put it in GLAD and it'll actually give you comparative visualizations such as heat maps. And also it has inbuilt filtering and sorting capabilities and data wrangling capabilities. And these in turn, you can actually see the structures alongside, provided the sequences are entered in a specific format. Even if the sequences are not in a specific format, you can still use GLAD to produce the data visualization and use the filtering and sorting capabilities. So the problems it helps solve is it helps visualize glycan structures alongside the data. It's useful for when trying to compare results from multiple experiments or samples, and also has a number of data mining functions which can help to filter and sort data. One more thing to keep in mind is this is a web-based application, so you can actually open the browser and use it, but none of your data is uploaded to any server. So it is kind of privacy friendly, because your data still remains on your browser only. It does not get uploaded anywhere. and everything is done on your computer in your own browser. But it also means that there is no saving on any server of the status of the application. So think of it like Word. At regular intervals, if you're using this application, I recommend that you use the save features in the software to kind of save your state of analysis at various time points, just so that, if things crash or anything happens, you don't start from all over again. So to give an overview of how GLAD works, the GLAD workflow starts with entering the data as tab delimited text files. And then this can be used to either visualize the data as bar charts, or this is then entered into the backend of the software as selections, which can then be saved or loaded, normalized, sorted, or filtered. And then this is the stage one of the visualization. But then once you have these selections, you can also generate the stage two of the visualizations, where you can use it to see data from multiple data sets, either as group bar charts, or as heat maps, or as network graphs or force graphs, or as calendar heat maps, or even produce correlation plots like this, where you can actually get values of how well samples correlate with one another. So just few important links to note. The main page is provided here. All of this is mainly on this glycotoolkit.com domain name. So as soon as you go to this website, you should be able to go to all of these pages one by one. And then lastly, I would like to point out to the publication where the GLAD was originally published. If you use GLAD, please remember to cite it. It just makes it easier. And also when you cite using GLAD, please mention the version number. I will also put in the version. I'll show you where the version number is. This makes it easier for reviewers or readers who come in later to your paper to understand what version of the software they had used. All right, so we'll move to the live demo. Just to give you an idea of what we will see in the live demo, we will see data for, actually it's 5 lectins, not 4. And in the bracket, these are the known binders for these lectins. So like AL binds to fucose, ConA binds to mannose, GNL binds to mannose, SNA binds to sialic acid, and MAL2 binds to sialic acid as well. These are the symbol nomenclature for glycans for these specific monosaccharides. So it just makes it easier to correlate the glycan names with the structures, and the symbols help in that. And then each lectin has data for 2 concentrations at 1 microgram per ml and 10 microgram per ml. And using this data, we will try to verify the known binding patterns for these lectins and see if we can dig in deeper for the data for this lectins. Okay, so with that, I will move to my browser. So hopefully you guys can see my browser window. If Renee, you can confirm. I think it should be fine. Okay, thanks, Yuki. So just to give you an idea of what we will go through. So I downloaded datasets for glycan microarray data either from the NCFG website, which is our website at the National Center for Functional Glycomics, or the CFG website, which was the Consortium for Functional Glycomics website. So the GNL dataset is from the Consortium for Functional Glycomics, whereas the others are all from the NCFG website. So let's just open the GNL dataset and see what it looks like in Excel. So in Excel, you can see that you have a list of sequences, the RFU value, the standard deviation, and the CV, and then the chart number. And you have the concentrations for 1 microgram and 10 microgram. Okay, so this is basically the data set which we will start off with, and then we will produce the input files for GLAD and go into the software. Now, before that, I just want to show you the pages again. Like if you go to glycotoolkit.com/glad in capital letters, it takes you to the home page of the software. This is basically giving you the description of the software, the latest tool version. So here is the version number of the tool. Also information of how to cite and the links to the documentation and the change log. The change log is basically when the version gets upgraded to the next version, the change log covers what was changed from the previous versions. The documentation, if you click on the documentation link here, it will take you to this page, documentation. It basically goes through one by one each of the features in GLAD. And at the end, there is also a complete example like what we are going to do over here, but it's a slightly different example comparing collecting data from the CFG. So, and within each of these sections, if you go in, you will have text documentation, but there's also small videos which basically shows you how you can use the particular feature of that documentation page. So this makes it a little easier for people to start off with just going through the documentation and learning about GLAD. Okay. And so this is the GLAD tool. Before we go in, let's check the documentation for the data format. So in the data format documentation file, you have the example data file structure. So you need a file in the tab text tab delimited text file to be saved in this format. So ID, name, RFU, standard deviation, CV, rank, and name too. So to start off, we have our data set from GNL. What I will do is I will just make a new workbook in Excel, and I will put it side by side. And then we need in this one ID, name, RFU, standard deviation, CV, rank, and then name too. And what I will do is I will copy this data from here and put it over here. And I'll just paste the values. So all of this is here. Now, the name column is where you want the sequence to be in the CFG nomenclature, which was the IUPAC condensed nomenclature. Now, if you don't have your sequences in this nomenclature format for the glycans, it is still okay. You can still use some other identifiers for your name, but GLAD uses this name format to draw the structures when you're visualizing the data alongside. So if it's not there, you might not be able to visualize the data, but you can still use all the other functionality of GLAD to draw heat maps or force graphs and all of those things. The rank needs to be calculated. So I will just use an equation to calculate the rank based on the RFU. Sorry. Let's do this quickly. And then we need to make this a constant. And then we will just drag this down. So this is an Excel function to just calculate the rank of the binding across the data set. Okay. And then the name too can be any other name for this which you would like to attach alongside this glycan. So you can have something like, monosaccharide and calculate them or enter whatever you want about specific glycan structure. So like monosaccharide and fucosylated. So some other nomenclature which you would like for the glycan can be entered in name too. This column is optional, so you don't need to have this. You can also just copy the the name into the name too. So you can just do is equal to this and then spread it down all the way. Okay. And once you have this file, this is your GLAD input file basically in Excel. You can then file save as, and I already have the demo file saved. But if you were saving it again, you just save this as a text tab delimited file. Okay, and you can give it a name which describes the sample, so GNL, and this is 1 microgram, for example, and save it as a .txt file. Okay, now I'm not going to save it because I already have prepared all those GLAD files for you. So we will not save this file, and I will just show you one of those GLAD files, right? So if I go into my GLAD demo folder, this is, for example, the AL Excel file. It's the same thing. It's the ID name, RFU, standard deviation, CV rank name too. And then this is the text tab delimited text file, which you can see here. It is the same thing, but it's just in a different file format. Okay, so once we have all our files ready, we can then start going into GLAD and using them. Now, just one more note. I know this is a lot of trouble to go through large data sets to convert this. So there is a tool called ExceltoGLAD, which also does this in an automated manner. It is just an Excel file with a macro. So you can download it and run the macro in the folder. There is a video guide to how to use it. But a note about ExceltoGLAD is it was built in Windows. I know people who use Macs have sometimes had issues with this. So you might want to use a Windows Windows computer to use this particular converter. Okay, so let's go back to GLAD and let's start using our data. So the first thing we start with, in GLAD, everything goes from left to right. So you start with use your own data, and we will use multiple files. So I can just click on multiple files, and then I can import these files one by one or all together. Now, by default, when we import multiple files, GLAD imports the files with the file name as the sample data. So when I import, for example, AL01, it uses the file name to give the data set the name of the this particular data. You can change the sample colors and names. So for example, if I wanted this to be, for example, 1 microgram per ml, I can just set that, and I can change the color also in case I don't like this green. I can change that. And now if I click on the individual bar charts, oh, I didn't actually change it. Now, if I click on this, it changes the color of the chart. I'm going to refresh the page and start over. When we import multiple files, GLAD automatically switches the data colors for each file. So it randomizes the colors just so that it's easier to visualize. You can click on the settings tab, and then you can uncheck this, and it'll only use this color for all the files from that point onward. Okay, so let's go ahead and import all the files. Okay, so I just selected all and imported it. What happens is you can see all the files are here now. So you have AL0 1 microgram, AL10 microgram, CONA, GNL at the two concentrations, MAL2, and then SNA is this. Okay. Now you notice that the order of concentrations is 0, 1, 10, 0, 1, 10, 0, 1, 10, 0. Oh, MAL, it switched for some reason. Now we want to sort the data so that the concentrations are all the same for all of the data sets. So this is where you can use the stage one, and you can sort the data sets. So let's get all the 0.1 microgram data first, and then the 10 microgram data after that. And what we're going to do is we're going to sort the samples by the data titles. Okay, so it gives you a prompt saying that it sorted so many data points. And then if we draw the individual bar charts, it has sorted everything. It has put all the 1 microgram data sets ahead of the 10 microgram data sets. The next thing we want to do is we want all the lectins together. So we want AL, then CONA, then we want GNL, MAL, GNL, MAL, and then SMA. Okay. So we did another sort. And now if we do the individual bar charts again, now we can see we have everything in the correct order, including the MAL2. It's become 1 microgram and 10 microgram. So this is the sort feature of GLAD. You can also sort the data by the glycan's name, which is the sequence, or IDs, and so on. Now, once you have done this manipulation of sorting, you want to save this sorted state of the data, and that's called the selection. So I'm going to save this sorted state of the data as a selection, and I'm just going to browse to the same location as my demo files. So let me just select that. There. And it's going to give me the option to save the select the file as the GLAD selection. And now, even if I refresh the page, I can just go to save load selections, go to browse the selections and load that selection file. And then you can load it to the current selections over here. Now, if I draw the individual bar charts, it will draw the same data set. So selections are like snapshots of your data, which you can save at any time of any point during your data analysis workflow. The next thing I want to talk to you about is, so you can also filter the data. Now, say we want to see the data for only fucose containing structures. But before that, how do we know what the structures are? So in this bar chart, if you mouse over on any of the data points, you can actually see the prompt or the tooltip which pops up, which shows you what the name is, what the name2 is, and the RFU is for that data point. If you look on the right side, there's this glycan structure drawer over here. If you click it, it pulls out a nice drawer over here, and you can mouse over any data point and it will actually draw the structures for you based on the name of the glycan at that point. This glycan structure drawer can be used in all of these visualization tools which are available in GLAD. And this is kind of where the power comes in to see, okay, this is binding. What is it really binding to? I can go in and see the structures, and it's immediately evident that these structures are all fucosylated, and that gives us the idea that AL is probably fucosylated. For reporting, you can obviously take a snapshot of this figure wherever you want. The other way to do it is you can hold down Control on your keyboard and click on multiple data points. And what it's doing is it's adding the structures to the selected structures window. And you can actually see the structures over here with the sequences and the suggested classes for those particular glycans. If you do not like your selections, you can clear all selections. and then start over within that again. Okay. Now, if you don't like the way GLAD is drawing this, I have another software called GlycoGlyph where you can copy these sequences and it'll give you more customization for that. That is also hosted on glycotoolkit.com, and you can paste it over here in that, and then you can also adjust multiple things like you don't want the text for the linkages to be rotated along the link. Maybe you want the abbreviated text format, and then you can redraw it and it'll give you like a better image for reporting. But you can always click on this camera button to get this image as a SVG file, or in GlycoGlyph also there's a camera button. So this view, which you see here, is called the individual bar charts for selected data. And what it allows you to do is it allows you to cycle through the data one by one. As you can see, I'm just going through the data one by one, and I can see all the data sets in this. You can also use this to highlight regions. So I can choose the highlight color. Let's choose a pink, for example, and reduce the opacity. And what I can do is I can use the scroll wheel to zoom into certain areas and then highlight specific areas where I see binding for AAL, for example. Now, these highlighted areas will remain for all the data sets. So this makes it easy for you to compare. Oh, okay, AAL is binding this highlighted area. What is CON-A binding to? Oh, it's not the same area. So it is much different glycans which is being recognized. At any given point, if you want to save the view of this visualization, you can click on the Save SVG button, and it'll allow you to download the SVG file. And that SVG file, you can again open it in Illustrator or any other vector graphics software and edit it over there. So say you don't like this way the RFU is, you can save the SVG file and then move this axis label over here or wherever you want. Okay, so that is the selections, the highlighting. You can also erase the highlighting by clicking on the remove highlight areas, and then you can start removing the highlighted areas. So that way you can highlight different things within this view. Say you don't like this dimensions of this, you can change the dimensions of this individual bar charts. You can change the X tick interval. Right now it's at 50, but you can change that to a smaller value or a larger value. And you can customize the y-axis. So say you want to fix the y-axis to 65,000, you can do that. So now all the plots which you see here, they will all be scaled to 65,000. And so this makes it easy to compare data sets where you see low binding or high binding for a particular data set. So you can see that some of the lectins, they bind low compared to the others. And so there is this option for normalizing the data. And what that does is it brings up all the values by default to 65,000. You can change this to any value. So if you want, you can represent it as a percentage. We choose 65,000 because that is the scanner settings, which are the peak scanner settings for the scanner which we use. And that's why that is the value over there. So if I normalize the data, what will happen is GLAD will pull this data to 65,000 for the tallest binder and then scale all the others accordingly. So when I click data normalization, it's normalized. And now if I click on the individual bar charts, you can see everything is up to 65,000. MAL2, for example, was a low binder. You can see that the top binder is now scaled up to 65,000. So this allows you to compare data from various data sets where the binding might not be at the same level for your data sets. Whenever you do any action in this in the data manipulations, an undo button is generated based on whatever action you have produced. So you can always undo your action for the previous manipulation which you had done. So if I do an undo right now, it undid the normalized data. And if I click on the individual bar charts, you can see that it's scaled back down to the normal scale of the binding for those particular data sets. Okay. All your data manipulations are saved in a data manipulation history. So you can go to this top right button over here and click on it, and it will show you what all you did. So the only step which I did at this stage was loading the selections. If I normalize the data again, and I click on there, it shows that the data is now normalized and the option was 65,000. Okay. So that's as far as the basic visualization is concerned, and also some of the data manipulations which we have done. Like I was saying, like we know that AL is supposed to bind all fucosylated glycans. So let's filter the data for fucose. So filtering data can be done by various methods. You can either filter the data by the glycan name or the name too, or by glycan IDs, or by the samples also. You can also filter by cutoff values, which are like you want to see only the results for the top 50% of the binders, or you can filter by rank based on the rank order which you have given in Excel. So let's do fucose over here. So I'm just going to type FUC, which stands for fucose. And if any sequence has FUC in it, it's going to filter the glycans for that. So it filtered 2004 data points. And now if I draw the individual bar charts, all of these structures are fucosylated. And if I pull out the glycan drawer, you can see that all of these structures have the red triangle in it, which is representative of fucose. What's interesting is that some of these fucosylated structures do not bind AL. So the blanket statement of AL binds all fucosylated glycans does not hold true, because it binds a majority of the glycans, but maybe not all. Now let's see what happens if we increase the concentration. We see that AL binds much more tighter to certain fucosylated glycans as compared to others. And the signal goes up as more and more lectin is on the array. In comparison, Conne doesn't seem to recognize all the fucosylated glycans because you can see a large chunk of these fucosylated glycans are not recognized. Let's undo the filtering and go back to the individual bar charts, and let's try to see what we can see for Conne. So for Conne, mostly it is binding to mannose type of glycans, which are these green circles. So we'll filter the data for mannose and see what happens after that. So filter glycans by name, and let's draw the individual bar charts. So now you can see AL is recognizing the mannose containing glycans, but only those which have fucose in it. But if you go to Conne, you can see that many more glycans are being recognized by Conne, and you can see the binding for those which have mannose. And if you go to a higher concentration of Conne, you can see much more binding for mannose containing glycans. But again, not all mannose containing glycans are recognized by Conne. Structures like this, which are different, they are not recognized as well compared to the traditional N-glycan containing structures. So this way you can go down and filter the data set to visualize more tighter or more refined sets of glycans within this. Now note that the x-axis is quite arbitrary in this case, because it takes it from the ID numbers, and because it's filtered data, it skips a few ID numbers, and therefore you can see that the scaling is off compared to the number of glycans which are there. Now, Once you filter the data, and if you want to filter the data in a much more refined manner, so say you want to see the structures only between 312 and 325, we can filter the data for 312 and 325 and say filter glycans by ID. And now if we draw the individual bar charts, it's only those data points from the filtered data. So we first filtered with manos, and then we filtered only the manos containing glycans between IDs 312 and 325. Now we have a small enough data set that we can see something like a group bar chart. So if you draw a group bar chart, you can see that this is the data for AL, this is the data for con A, and so on. Now it doesn't help that all our samples are of the same color. So let's go and change the sample colors. We'll give con A a pink, okay, GNL a purple, okay, MAL2 maybe a green or a teal, and then let's give this let's give this a brown or maroon. So once we have that, if we draw the group bar charts, now you can see that you have a figure where your data, your legend is showing you the color of the chart, the bar, and it tells you which lectins are recognized by these small set of glycans over here. In this grouped bar chart, it takes the x-axis just based on the number. So 1, 2, 3, 4, 5. And within one, you can see the data for all of these binders over here. Okay. And again, in this also, you can mouse over the data points to see the structures for these glycans. I'm going to show you the next thing, which is the heat map. So you can choose the size of the heat map grid, which is the squares it draws for the heat maps. And there are various color themes which you can use. By default, this is the color theme which is there. And then if you click on the heat map, it will show you the binding for these specific glycans, which we have filtered down to. And the color of the heat map reflects the binding intensity. Now notice that there are some of these glycans which are not there for all data sets. Like GNL, for example, does not have this set of glycans, and this set of glycans is extra in GNL, which are not there in the other data sets for the NCFG. So there is this function called discard unmatched data, which will go through all the files which you have imported into GLAD, and any data point where the sequence of the glycan is not there in all your data sets, those data points will be eliminated. So this way it makes it easier to compare data across data sets where you do not have the exact same glycans printed. So let's do a discard unmatched data. And it says could not find matches for 5 glycans in all samples. So it's going to remove these 5 glycans from the data set. And now if I draw the heat map, it's reduced the number of glycans, but the data set is more comparable across the board because all the glycans which we have in this data set are now there in all the samples. So this allows us to draw better conclusions across data sets which might not have the same types of glycans printed on the arrays. Like I said, you can scale it down or up. So let's say we don't want the scale to go to 70,000. Let's change it so that 2,000 is the minimum. You can make it 5,000, and let's make this 10,000 and 20,000. Okay. So this basically shows you the scale. And now if I draw the heat map again, it shows you the new scale and the new the new figure. Now, if you want to highlight only the rows and columns of this, you can click on any of these cells and it will highlight that particular cell for you. So this makes it easier to drive into your data and kind of remove the noise when you want to see the data for a specific region of your data set. If you want your samples to be as the rows and your glycans to be the columns, you can click this rotate heat map button and it will basically rotate your heat map. And you can click and drag this around to adjust it relative to your key, which you can have alongside. Again, glycan structure, you can see at any given point. Control clicking will add those structures to the selected structure window. Oh, yeah. Okay. So that's the heat map. When you have data, which is, I'm going to reload all the selections. Okay. So let's load the selections again and start over. Okay. So when you have a large data set, drawing heat maps becomes difficult. So this is the full data set with 600 glycans and the heat map is like really large. So this makes it difficult for you to draw heat maps. Same with individual bar charts, it becomes difficult to show all of these bar charts alongside. So calendar heat maps is a visualization which we have come up with to help with that. So what it does is it starts putting all the glycans one as a heat map in the row, but each sample is represented by this one block of the heat map. So you can compare the samples across multiple of these blocks. Now again, because the data set is not identical, you can even see which glycans were missing from this data set compared to the other data set. Or you can use the discard unmatched data to remove this and remove any other glycans which are missing across the data sets to get a more uniform calendar heat map. Force graph is something interesting. So force graph, what happens is I'll first draw the force graph and explain what it does and how it's generated. Okay. Force graph. We should change the colors back again. So Connie, let's make it pink. Let's make this purple. Purple. Let's make this pink. Let's make this. And now I'm going to save the selection again. Just overwrite my existing file, just so that I can come back to this again. All right, so let's redraw the force graph, right? So when we redraw the force graph, what it is doing, in this case, what it does is each of these colored nodes represents your sample. So for example, these brown colored nodes represents your sample, which is SNA. Each of the gray nodes represents a glycan structure. So all of these gray nodes are glycan structures. So this allows you to see what your samples are binding to. And if you have multiple samples, you can find out common binding partners for 2 samples or more samples. So if you click on this node, double click on one node, you can kind of see which binding partners recognizes this specific glycan. And this is the power of force graph. Now, the distance the node takes from the colored node, the glycan node takes from the colored node, that is dependent on the strength of binding. And so it tries to always pull the nodes in such a way that the force is directed by the binding intensity. Now, if you change some parameters in the force graph, like the minimum and maximum or any of these parameters, the figure which is generated by the force graph will change. And so it is important that when you see a force graph, you should know what the parameters were to generate it. Otherwise, you will not be able to generate it again. In the force graph, you can highlight nodes using the same name nomenclature. So if you want to see all the structures with sialic acid and you wanted to give it a highlight of orange, you can click on type this in and say go, and it will show you all the glycan structures which had sialic acid. If you want to clear it, just clear this and say go, and it goes away. And you can take this photo as a SVG again at any given point, and you can pull out the glycan structure drawer to see the structures if you want to alongside the data for this. Now, if you want to change this scale, you can reduce the minimum binding to 500. That will keep the binding lower. Or if you want to see only the top binders, you can increase this and the number of nodes should decrease. But again, like I said, the force graph will also change slightly. If you feel that things are just going towards the edges, you can increase the force strength and the link distance or decrease the link distance to bring things closer or far apart as you want. Okay. Correlation map allows you to quantitatively see how similar two samples are. So if I click on correlation map, it draws this map. And this uses Pearson R correlation to calculate the R value between two samples. So for example, this particular cell has a correlation R value of 0.878, which is over here. And it's to be expected, right? AL is the same protein sample, it's just at two different concentrations. So either 1 microgram or 10 microgram. And the R value for this, both of these is about for this comparison between this versus this is 0.878. So using this correlation map, you can see how similar two samples are. Like GNL and CONA are probably more similar because they both recognize mannose, but not very similar because even though the R value is still very low at 0.187. And if you see the data set, you can find out what are the common binding partners between the two samples using this. Okay. And for the correlation map, also there are color themes which you can do. There is a bland ointment plot, which is just another way of drawing the scatter plot, which allows you to see which glycans are out of the standard deviation, and you can adjust those values over here as well. And also the range of the color scheme which you want. So maybe if you don't want to go to -1, you can go from 0, 0.25 to 0.5 to 0.75 and 1, and redraw the correlation map. And now you can see more. It makes things more prominent to see the more similar binders as compared to the non-binders. And the last visualization is the bubble box plot. In this, each glycan is represented as a dot on this scatter plot, and then each of these on the x-axis is the samples for each of this data set. So if you go pull out the glycan drawer, you can hover over each glycan and see how it binds relative to all the samples. And you can click on it and it'll highlight it and leave the highlight so that you can take a picture of this plot, for example. You can also spread the nodes a little bit so that it's easier to see all the glycans. You can use something like a spray plot also to spray out the glycans so that the top binders are more spread out because the lower binders might not be as interesting. You can draw violin plots with this. And you might want to log transform the data when you draw the violin plots. And you can also draw box plots to see which glycans bind more or less. So usually I like to use bubble box plot along with filtered data. So if I say, okay, fucose, let's filter all the glycans by fucose. And then you can click on bubble box plot and then draw the boxes, and you can see that yes, AL definitely significantly recognizes more than the others. And you can do an ANOVA analysis also to see which binds more or which is significantly different binding compared to the other using this. And you can group the samples together also to get more aggregated data using this group feature. So because time is limited, I will not go over that feature. But basically, this is an overview of GLAD. Now, we made several changes to the settings also. So I showed you how to save selections, which is a snapshot of your data. But if you want to save this, the whole snapshot of your session, you can save it and load it using these two buttons over here. So you can save the session using this button and it'll save it as a GLAD session. And then if I open another GLAD window, for example, I can load that session back. And what it will do is it will not produce all the figures, but it will keep the settings what we had changed. And that will allow us to redraw it by just clicking on the course graph or calendar heat map. And it also records your data manipulations when you save the setting. So that way you can even undo the previous data manipulation and get back to the original data which was there. So that is the best way to report a GLAD session to somebody else who might want to take a look at or report it in the journal article would be to save a session and just attach that as a supplementary information for the reviewers, and they can use this software to visualize it also. And lastly, again, remember that the version number is also at this bottom right corner. So you can note that down when you're reporting the data as well. Okay, with that, I would like to stop the recording, and also I think that is all I would like to say about GLAD. If you have questions, you can we can continue after this, and thank you.